Today, what this video is going to cover is a type of genetics called additive genetics. Additive genetics is something that we study in the broader category of what's called quantitative genetics. Where quantitative genetics, we're interested in studying the underlying genetics of complex physical or complex traits. Um, Easy example to think about is human height, in which we have a wide range of possibilities of human height rather than specific categories that we fall into. People don't fall into 5 feet or 6 feet. We could fall anywhere in between, or even shorter or taller. Okay, so these complex characteristics or these quantitative traits, they tend to be what we call multifactorial, and that multifactorial means that they're polygenic, that they have more than one gene contributing to them, and that those genes interact with the environment to give you your final phenotype. Now, one aspect of these genes is that when we consider genetic traits, there are different ways that genes manifest into the phenotype, that genotype to phenotype relationship. And so, one is what we study when we look at Mendelian type P crosses, and this is a dominant recessive type relationship. Another type of relationship is what we call gene interaction, in which different genes will interact with each other in different ways, and different combinations will give you different outcomes. And then the third type of genetics that we consider is what's called additive genetics, in which different genes will add to some base height or base trait. And like if we're talking about human height, that there'll be kind of some basic height that humans will tend to be at, or a plant or something. And then you'll have multiple genes that can add to that, and each of those different allelic variants will kind of add different amounts. The example that we're going to look at today is a really simplified example of this, and we're only going to be looking at the additive aspect of this. And it's a really kind of simplified version. When we look at human height, we know that there are probably over 70 different locations in the genome that contribute to human height. Some of these locations contribute just a few centimeters, some could contribute up to an inch or so, depending on which variant you got. And there, in each of those different 70 locations, there are probably multiple variations of those genes or multiple alleles. And so what we're going to look at today is an example that is really simplified, in the sense that we're only going to consider that we have two genes contributing to the height of these individuals, and that each of these genes only have two alleles. But it's a useful kind of framework for us to work with in terms of how additive genetics will work, and how we can kind of add to, depending on how many of these different allelic variants you get, you're going to actually get you'll get different phenotype outcomes, but you can have the same phenotype with many different genotypes. Is the really kind of the take-home message that we want to see from this worksheet, is that there are multiple ways and multiple combinations of different allelic variations that you can combine in different ways that give you the same phenotype. And that's ultimately what additive genetics is going to show us in terms of at least this type of a problem. So this is a kind of a spin-off of a based in fact, but really distorted over time now. There was a king in Prussia. His name was not Frederick Colling. There was a king in Prussia who wanted to have really large guards and tried to set up some sort of breeding program to get really large guards. He wanted to be known as having the largest guards. And so this is kind of a spin-off of that. And so what this prompt is telling us is that he spent 20 years trying to having giant humans with members as tall as 7 feet as part of his personal guard. And he did resort to bribery and kidnapping and smuggling to try to fill his unit. Now, so what we're going to look at here, is, so assume that that height in this case is determined by two pairs of genes. Okay, so with any problem like this, and they're all going to be set up exactly the same in terms of just trying to work out additive genetics, and we're going to approach solving them exactly the same. There's always going to be a minimum value. There's always going to be a maximum value. And then we'll have some indication of telling us how many genes we have. Key thing to remember when you have how many genes you have, we're going to be told in this type of a problem, it'll be 2 different allelic variants for each of these genes. One will add to the trait, one will not. Okay. And so we can denote the ones that add to the trait as being capital letters and the ones that don't as being lowercase. We could denote them as a plus symbol if it adds to the trait, or a minus if it doesn't. But will always be 2, for this type of a problem for us, there'll be 2 different variations of each gene, or 2 different alleles, and you'll have a minimum value, a maximum value, in this case height. We'll have a minimum height, a maximum height. We're told that we have 2 genes, so we have 4 alleles. Okay, it could be length, it could be width, it could be diameter of some organism of whatever trait, and it could be 5 genes, it could be 4 genes, it could be 2 genes, it could be 6 genes. However many genes you have twice as many alleles. Okay, so for this case, we have 2 pairs of genes, so we have 4 alleles. The homozygous recessives, so what we're referring in terms of this, homozygous recessive neutral genotype, in which we have no additive alleles being added to this trait, is 5 feet. So that's our base height. Okay, all individuals in this problem will be at least 5 feet tall. And then depending on how many more additive alleles that these people get, will add to the total height, that where the tallest individuals are going to be 7 feet tall. Okay, so now the first question that we need to answer is how much height is added to that 5 feet for each additive allele. So for any problem like this, doesn't matter what we're measuring, okay, it doesn't matter how many genes we have, what we're going to do is we're going to find the difference between the maximum value and the minimum value, and we're going to divide that difference by how many alleles we have. And in this case, we're going to have feet, or we can consider this in inches. We'll have so many feet between the maximum and minimum, divided by our total number of alleles. That'll tell us how many inches per additive allele is going to be added with each of these additive alleles. And so what we'll do here is we have 7 feet minus 5 feet is going to be 2 feet, okay, which for the sake of, we'll convert to inches, so we can talk in terms of how many inches each of these different additive alleles adds to our base height. And so if we have 24 inches divided by 4 alleles, what we get is 6 inches per allele. So now we get really hypothetical, kind of more storytelling. As it turns out, one of the tallest guards had an attraction for shorter women. And so he was 7 feet tall, and he was attracted to this 5 foot tall woman, and they had several different children from this clandestine affair. What were the genotypes and phenotypes of the F1 children? So if 7 is our maximum value, and if we're, say, we'll denote additive alleles as capital letters in this example. So the guard genotype would say capital A, capital A, capital B, capital B, because he had 5 feet. He had all additive alleles. Each of these additive alleles are adding 6 inches. So here we have 6 inches, 6 inches, 6 inches, 6 inches. So altogether 2 feet. That gets him to 7 feet. Is being crossed with a female that is 5 feet. So she has no additive alleles. So her genotype is this. And so the F1 would be all heterozygotes. Okay. And so the question is, what are the genotypes and the phenotypes of the F1 children? And so all of the F1 children are going to be heterozygous for both of the genes. And so that means that they have the base height of 5 feet, and then they have 1 and 2 capital letters or additive genes. So for each of those additive genes, we're going to add 6 inches, like we just saw above, like we just calculated above. For each additive allele, we're going to add 6 inches. So we have 5 feet 6 inches, and then another 6 inches gets us to 6 feet. And so all of these individuals are going to be 6 feet tall. Okay. So now, subsequently, some of the F1 children who had no way of knowing they were related married and had children of their own, give all possible genotypes and phenotypes for the F2 progeny. So this gets to be a bit of a long answer, but it's worth kind of walking through. And so I'm going to get some blank area here just to kind of give us some space. And so what we're doing is we're crossing an heterozygote for both traits with another heterozygote for both traits. So now these are independently, we're not told that they're linked, but in the problem we can just, since we're not told any information, we'll assume that they're unlinked. And if they're unlinked, then they're sorting independently, and we can determine probabilities completely independently of each other. And so what that means is that first we can kind of work out a quick little Punnett square. This is going back to the things we've talked about in terms of Mendel's P's. Oops. In which we're going to consider the possible outcomes in terms of genotypes for the A gene and in terms of the B gene. Okay, so we have 1/4 of the children are going to have capital A, 1/2 are going to have capital A, lowercase a, and 1/2 are going to have lowercase, and then the same thing for the B gene. Okay. So we have 1/4 capital B, 1/2 capital B, lowercase b, and 1/4 lowercase. Okay, so now if we want to take a look and remind ourselves what the problem is asking, okay, give all possible genotypes and phenotypes for the F2 progeny. So now what we need to do is we need to figure out, all right, what happens in terms of what are the different combinations we can get. And so for all of these cases, so for capital A, we could get a capital A with a homozygote dominant, with a heterozygote, or with a homozygote recessive. Likewise, we could also get this heterozygote going with either the heterozygote, or homozygote, and then here we could also get this homozygote recessive, could end up with each of these possibilities. Okay, so now, how we would kind of depict this is, again, just regular old probabilities, Mendelian cross type problem here. We can combine our probabilities. What is the probability of getting a homozygote dominant? And so we can use the fork line method here, in which we have 1/4 possibility of getting big A. Oops. And this could be with a 1/4 probability of big B, a 1/2 probability of big B, little b, and a 1/4 probability of little b, which means that for this first step we would have a 1/16 of the offspring would be homozygote dominant. Then 1/4 times 1/2 is 1/8. So let's just keep things in terms of sixteenths. 1/8 is 2/16 of the offspring would have capital A, capital B, lowercase b, And then 1/4 times 1/4, we'd have 1/16 of the offspring having capital A, lowercase, lowercase b, lowercase b. Okay, and we can do this across all of them. So 1/2 big A, little a, and we get 1/4 big B, 1/2 big B, little b, 1/2 little b. And so we'll go back. Right now, all we're doing is calculating the genotypes. We can then go back and translate these into what the phenotypes would be. And we can also then figure out what ratios we have. Okay. So now 1/2 times 1/4 is going to be 1/8. 1/8 is 2/16 of the offspring are going to be heterozygous at the A locus, and then homozygote dominant at the B locus. Here we have 1/4. So 1/4 is the same as 4/16. And so we're going to have A, big B, little b. And then here, 1/2 times 1/2 is going to be 1/4 as well. Oops, that's wrong. This is actually 1/4. So that's 1/8 or 2/16 are going to be big A, little b, little b. Okay, and we got one more to do. Okay, so now what we have left to do is, mistake there, it's 1/4. Okay, and so now what we have to do is combine the little a with all of the possibilities for b. And so in that case, we're going to have 1/4 little a crossed with 1/4 big B, 1/2 big B, little b, 1/4 little b. And so cross and cross. And so this is 1/16 will be little a, big B. We will have 1/4 times 1/2. 1/4 times 1/2 is 1/8, which is the same as 2/16. We're going to be little a, big B, little b. And then this is going to be 1/16 is going to be little a, little b. So now let's convert these into actual height. So for this first one up here, that's 7 feet tall, okay? Because we have the base height of 5 plus 1, 2, 3, 4. Each of these are going to add 6 inches. So the A locus is adding a foot. The B locus is adding a foot. We have 5 feet. At the other extreme, we have that base value of no additive alleles. So this person is 5 feet, okay? And then for this, we have 2 out of 16 are going to be little a, big B, little b. So this is 5 feet, and then there's one capital letter. Doesn't matter where the capital letter is. For every capital letter, you're going to add 6 inches, okay? And so all of these 2/16 people are going to be 5 feet, 5.5 feet, okay? Now this 1/16 of the people are going to be little a, capital B. Here we have 2 capital B's, each of them adding 6 inches. So we're going to add 6 inches and 6 inches, or a foot, onto our 5 feet base. So all of these individuals are going to be 6 feet tall. Now if we look up here, we have one additive allele. One additive allele gives us 5 feet 5 inches. Now here we have 2 additive alleles, just like we have 2 additive alleles down here. It's going to give us 6 feet. Here we have 1, 2, 3 additive alleles. For each additive allele, we're adding 6 inches. So now we're going to add 1.5 feet to our base height of 5. So now we have 6.5 feet. Okay. Here we have 1, 2 additive alleles. So we're going to add again 1 foot. So these are going to be 6 feet. And then this one we're adding 1, 2, 3. So we're going to have 6.5 feet. So what we can clearly see here is that we have multiple different ways that we can get to having the same phenotype. Okay, because here we can have 6 feet individuals by having this particular genotype. We also could have 6 feet by this particular genotype. We also could have 6 feet by this particular genotype. Okay, and so when we look at phenotypic ratios, okay, we have, for possible phenotypes, we have 7 feet, we have 6.5 feet, we have 6 feet, we have 5.5 feet, and we have 5 feet. In terms of ratios, well, of all of these individuals, we have 1/16 is going to be 10 feet tall. Okay. And then 6.5 feet tall, we have this possibility of 6.5 feet, and we have this possibility of 6.5 feet. So we have 2/16, and then we have 2/16. So we have 4/16 are going to be 6.5 feet. Okay, and then for 6 feet, we have this possibility, that possibility, and that possibility. So we have 1/16 plus 4/16, that's 5/16, plus another 1/16. So now we have 6/16 of these are going to be 6 feet tall. And then 5.5 feet, we have this possibility and that possibility. So 2 and 2. Here we have 4/16. And then for this last one, we have 1/16. Now we should add up to 16 here. Okay, so here we have 4 and 6 is 10, plus 4 is 14, 15, 16. 16, 16. So those are all of our individuals. So phenotypic ratio, we're going to have a 1 to 4 to 6 to 4 to 1 ratio. In terms of genotypic ratio, you could say that, you typically wouldn't say this, but you could say that we have a 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1 ratio. This would be more likely what you're going to see in terms of phenotypic ratios, but we have multiple ways that we can get to that particular, to those different phenotypes. And so we have that 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1 is what we could say. For genotypic ratios, here are phenotypic ratios. Now, so that is how we would go about answering this next question here. Is in terms of phenotype, what we would have is we'd have 7 feet at a 1/16. We'd have 5 feet at 4/16. We'd have 6 feet at 6/16, and then we have 5 feet 5 inches at 4/16, and 5 feet at 1/16. So this would be 1 to 4 to 6 to 4 to 1 phenotypic ratio. Okay. Now, possible genotypes we just wrote them out on the side. I'm not going to rewrite them all, but that was the genotype ratio. If we wrote it out, would be 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1. Okay, but that is reflected in what we did here. Okay. All right. So now the next question for this problem is, what would the average height be? Okay. So now there's 2 different ways you could go about doing this. Okay. So one is we can just calculate 7 feet. Okay. So that 1. We're going to add to that. We got to add up all of these different heights. 6.5 times 4 plus 6 feet times 6 individuals plus 5.5 feet times 4 individuals plus 5 feet times 1 individual equals, and now we have, so 96, and then we have 16 different individuals divided by 16. So what would the height be? What would the average height be? The average height would be 6 feet. Okay, so now I said there are 2 different ways to do this. So that's one way of just calculating it out, is just to calculate out exactly how many differences there are. The other way to do this is to remember the work by Edward East. He's the guy that worked with tobacco flowers. And what he found is that when you take a quantitative trait and you start with a p generation, that is your 2 extremes, and you have those 2 extremes, that in your F1 the height is going to be, the average is going to be in the middle of your 2 extremes. So in this case we have 7 feet and we have 5 feet, so the F1s, they were 6 feet, right in the middle. Then what he found was that when you go from the F1 to the F2 is that the mean stays the same. The variance gets much broader, okay, but the mean stays the same. So when you go to F1 to the F2, you should have 6 feet as well. And so mathematically we just worked that out. Here we have we're going to have 6 feet because we have 7 feet times 1 plus 6.5 times 4 plus 6 times 6 plus 5.5 times 4 plus 5 times 1 divided by 16, or we remember the principles that Edward East found, is that it should be 6 feet, and mathematically it works out. Okay, so now if these individuals had then in turn had children with the general population of the area who had an average height of 5.5, what would then be the average height of the children if there were no environmental influences? And so what this means by no environmental influences, it means that if we're talking in terms of broad sense heritability would be 1 because there would be no environmental influences at all. And show your work by either listing all the crosses and frequencies or using quantitative genetics. So we could use quantitative genetics here, in which we can calculate out our average height by calculating the average height divided by times broad sense heritability. So in this case, again, or we can just use Edward East. And when we consider Edward East, okay, if we have 2 extremes of 6 feet and 5.5 feet, okay, then what we should get, and then we have no environmental influences at all on this, what we should be getting if we have 6 feet and 5.5 feet, 5.5 feet. So what we should get is, if we add that and divide it by 2, if we just take the average of these, in which case we have 6 plus 5.5 divided equals 6 plus 5.5 equals divided by 2 equals 5.75. And we're going to, this is a big simplification, but this would be using Edward East's principle and showing that the the average height, since the average height of the population is 5.5 feet. You take an average height of individuals that are about 6 feet, crossing them, they should have, those are your two extremes. The mean should be, the average height of any offspring from that should be in the middle of those two extremes. Okay. And then, so here this is just kind of going on the story as a little bit of a thought problem. And so so this didn't work very well for the king of Prussia. He tried and it didn't quite work. And so the question is why. And the reason for this is because it's a lot more complicated than two genes here. And I'm not going to write out an answer for this, but it's more complicated than just two genes. That we have those 70 different genes, which means that we have many different ways that we can get to getting to that 7-foot mark. And so for all of those 7-foot tall guards that he had, or 7-foot tall women that he was having them have children with, they would have their height alleles, their additive alleles, would be probably heterozygous at a lot of those different locations. And so you'd still kind of get this averaging down a little bit. It's what we call a response to selection, which is something we'll cover in one of the next videos, is where you don't tend to get as big of a product as you get, even though they're your biggest, you still have those shorter alleles within those individuals because they're part of the population. And so this is what we would call a response to selection, in which you don't get as extreme as the extremes that you take, in this case 7 foot and 7 foot. We'll look in another video at an example of response to selection. Would be kind of the reason behind that. And then would this have worked if he had brother sister matings? Lots of ethical issues with that, but if that were the case, yes, this would probably have resulted in a better outcome, in the sense that he would have obtained taller guards. Controlled matings and stuff like that obviously is not good in any scenario, but he would have succeeded in his desire to have tall offspring, because all of those brothers and sisters, they would have been their tall alleles would have been at the same loci, and so you would have enriched for the probability of getting homozygote being homozygote at those locations. These two problems, just a little bit of thought about it. More importantly, for this sort of a problem, is to understand the additive genetics component of how do you figure out what your phenotype would be given so many genes with so many alleles in your two extremes. Okay, and so with that's how we kind of work out an additive genetics problem. Hope this was helpful. And we'll, in another video, we'll look at some more quantitative genetics type questions. Specifically, we'll look at a response to selection, and we'll look at another simple example of this sort of an additive genetics. Have a great day.